submitted our Medicats job. Let's see if it's done, the one that we submitted with feature groups. So you can get into your jobs by going up here and clicking on My Jobs. You can also get it down here at this lower right corner. Let's go here to My Jobs. And this is the one that I just ran, which was amoxicillin PBP2X. And I was comparing the susceptible and resistant groups. So let's look and see what that job looks like. Up here, this is Where Am I? This is the name of the job. This is the folder within a folder within my workspace. This is the information on how that job ran. This is the unique identifier for that job, the job ID. When it started, when it ended, a total of 14 seconds once it runs. Some of you will look at it and say, hey, I had to wait an hour for this job to complete. That's because there's a queue and it's a fair queue, so we all have to wait our turn. If you need to see what you submitted, you can click there and open the parameters box, and it tells me that I submitted these two groups, the resistant and then the susceptible, and what the p-value was for those. Now, from the introduction, we described what each of these were. So I'm going to start at the bottom and work my way up. The output is just an alignment file. And note that I could create an MSA for it right here. But let's look at it first. And it loads it, and this is just the alignment file that it's using. For those of you who don't know, it's got a top header with the caret in front, and then the next is the sequence. So this is the sequence for that particular protein, and then it starts with the next one. This is the genome ID of the genome that this protein is in, and this is telling me PEG stands for protein encoding gene, and it's number 331 in the way this thing was annotated. Okay, so I can go back up this way to the job and the breadcrumb. The math log is showing how the alignment ran, and kind of boring. I mean, sorry, for those of you who like this, it's very exciting. For me, it's kind of a boring file. So it's just showing you how the pipeline ran. Go back up. This is the MC table, which shows the multiple comparison p-value. This isn't the one I generally like to look at the most, though. So I'm going to go back up here, and I'm going to click on the chi-square table. This is the one I like to view. So I'll click on that. I'm going to give it some more real estate. And these are not all the positions in this alignment, because there were 750 of them, but it's showing you the 85 that were the most significantly different. Now, in the publication by Stanhope, they talk about the amino acids that are the most critical for resistance. So let's look at those. And the first one is position 339. Oh, too bad. We don't see a difference there. So sad. But the next one is 371. So I can click on that, and I can see that they found a significance difference. Here's the p-value here between the resistant and the susceptible proteins. I'll call them 2X proteins because I'm sure you're tired of hearing me stumble on PBB2X. Note, though, that some of the susceptible have the amino acids associated with resistance. Now, Stanhope et al. Don't say that all of the proteins have all of these mutations. What they're saying is that each of these locations that we're discussing, resistance had a particular phenotype and susceptible had a unique one. So it could be a combination, but certainly not all of them that are responsible for resistance. So here's 371. The next one is 384 that's described in that paper. And you can see that all of the resistant ones have that particular amino acid. And some of the susceptible have it, but most of the susceptible have an arginine there. And then the next one is 400, which we don't see here. So we have to go to 514. That was the next one. Now, this one seems pretty evenly split. Some of the resistant have the resistant amino acid, which is a histidine here. The others don't. And a minority of the susceptible have it. So this is what makes antibiotic resistance such a puzzler. You want it to be a, slam dunk, that if it's got this mutation, they're all resistant, but it just doesn't work that way. And also there are different proteins here. So if you knock out PBP2X, or if it's impaired, one of the other penicillin binding proteins can help the cell complete that outer membrane, perhaps. So there's a bunch of things going on in this organism, in this war against the antibiotic. All right, the next one is 546. This one also has a significant difference. Valine is the amino acid associated with resistance, and all of our resistant ones have it. And a minority of the susceptible, most of them have the leucine. And then the last position is at 605. You can see the significance here. And the resistant position is the T, which some of the susceptible have. So it's not clear cut, but there are statistically significant differences at these locations. So if you were going to report it in your paper, you could give the p-values associated with this based on the groups, and noted that we didn't have to have a one-to-one correspondence in the group because there were many fewer resistant 2X proteins as compared to the susceptible ones. So this tool is very good for that. I could copy the rows, and I could copy the rows I selected with the headers, so I would have that. And then I could paste it into an Excel file, and I would be ready to go. Excel is the biologist's friend, so I'd be able to isolate it, document it, and look at it there. So those are the job results for the chi-square table. Let me show you one more thing on this. I'm going to go up here to Medicats. I'm going to go to this job. When I use the breadcrumb, I lose the reference to this guy. If you click on the view here, it will take you to that chi-square table, so you don't have to deal with all of the folders underneath. So that is the job results for the Medicats service in a nutshell. In the next videos, we'll talk about the different submissions that you can do, either an auto grouping or from an alignment file. Thanks a lot. Bye.